September 22, 2017. A research led by Jordi Surrallés, professor of the Department of Genetics and Microbiology at the Universitat Autònoma de Barcelona, director of the Genetics Unit at the Hospital de la Santa Creu i Sant Pau and lead researcher at the Center for Biomedical Network Research on Rare Diseases (CIBR), has identified a new genetic syndrome caused by mutations in both copies of the FANCM gene, also known as biallelic mutations. The results, published in Genetics in Medicine, the official journal of the American College of Medical Genetics and part of the Nature Group, suggest that these mutations predispose the body to early formations of tumors and chemotherapy toxicity. In the article, lead author Massimo Pagliaiolo from the CIBR research group led by Jordi Surrallés analyzed biallelic mutations in the FANCM gene in 3 individuals. Despite the number of patients always being low in these types of studies, given that they deal with rare diseases, it was observed that there was an early onset of cancer and toxicity to chemotherapy, but the patients did not present any congenital malformations or hematological phenotype which could suggest being affected by Fanconi anemia, a rare disease which affects 1 out of every 100,000 children. Until now it was believed that the FANCM gene was related to this disease, given that in 2005 the biallelic mutation was observed in patients suffering from Fanconi anemia. In another article published in the same volume of the journal, researchers from Dr. Surrallés' group and the research group led by Javier Benitez at the CNIO in the CIBR confirmed that women with biallelic mutations in the FANCM gene did not develop Fanconi anemia, but did present a higher risk of breast cancer, chemotherapy toxicity and chromosomal fragility. This article was coordinated by Paolo Longo of the Milan Institute of Molecular Oncology, and included the participation of several hospitals and research centers of Italy, Germany, Spain, and Sweden. Until now it was thought that biallelic mutations in the FANCM gene caused Fanconi anemia, but we have now demonstrated that it is not so, given that in the 2 studies there were 8 patients with these mutations, and none of them had anemia, affirms Jordi Surrallés. The patients, however, had suffered from cancer at very early ages, and also presented chemotherapy toxicity. Therefore, in view of the new syndrome, the authors recommend modifying the clinical monitoring of patients with biallelic mutations in the FANCM gene and taking precautions when using chemotherapy and radiation therapies due to the acute toxicity they may produce. Genetic complementation test. For the study, researchers conducted functional complementation tests, a very important type of analysis in mass sequencing projects in which there are several mutated genes, and it is not clear which ones are the source of the disease. The patient cells have a clear phenotype of chemical hypersensitivity to DEB, an agent which causes damage to DNA. Patient cells do not survive high doses of DEB. In contrast, when a healthy FANCM gene copy was transferred into the cells of patients with the help of a virus using lentiviral transductions, researchers observed the reversal of this phenotype in cells behaving as if they were healthy, a response similar to that of a healthy donor. This functional study is the genetic demonstration that the gene causing the disease is FANCM, and, therefore, the mutations observed in this gene are of a pathogenic nature. And Universitat Autònoma de Barcelona. Related cancer articles. Current cancer news and events.